Welcome, everyone, to today's virtual talk. It's a pleasure to introduce my colleague and friend, Gordon Southam. Gordon is currently a professor at the University of Queensland. He has a joint appointment in the School of the Environment and the Sustainable Minerals Institutes, and has done a lot of work in both places, looking at very academic research about geomicrobiology and also increasingly in recent times, very applied research about how we can use microbes to help us with different problems involving minerals. Gordon comes from Canada. He has a bachelor's degree and a doctorate from the University of Guelph in Canada. And today he is going to be talking about geomicrobiology in general and giving us a tour through a variety of his research, which will range from looking at iron deposits to gold deposits to how you can increase weathering, to store carbon, to how you deal with acid mine drainage, a real tour through things. So I think we'll get a taste of these different aspects of geomicrobiology. And at the end, there'll be some time to ask questions.
For the presence of ore at or near the surface. Now, if you look at this material as a microbiologist would, again, zooming into the micron scale, we find that it's full of bacterial fossils. So in actual fact, the Romans were tracking the bacterial fossils in the creek up to find the ore bodies. And so in this view, we're gonna go back and forth between the idea of bacteria being good or bad. And we're not in the medical field, so where a lot of bacteria seem to be bad. We're in environmental bacteria. And so we want to ask the same kind of question. So here's the copper and mine tailings in the province of Quebec in Canada. And this is one of the first mine tailings that I ever went to. And as I was standing there taking this picture, I was told that that the generation of acid and the growth of iron oxidizing bacteria was impossible within this material. And what they were referring to is that the mineralogy here included calcium carbonate. So the calcite is a buffer, and so it should have stopped the iron oxidizing bacteria. But you can see the rusty spots here are almost a meter in size. And what in fact happens in these environments is the carbonates become coated with rust. It passivates the surface, they stop generating acid, and then the other minerals that are in there, like pyrite, become colonized, allowing for the growth of bacteria within this environment. So the chemistry that the bacteria is seeing are different than the bulk chemistry that we would look at this tailing at as a geologist would. Over time, as little as kind of 3, 4, 5 years. Mine tailings will go from not acid generating to acid generating. And what happens there is that the rusty spot here that you can see where I've kind of kicked the surface of the tailings, in fact, extends over this entire tailings. And so when you come out of winter, things are cold. And once the biology picks up in summer, the bacteria generate acid, and you have acid mine drainage that's leaving these sites. And so the legacy of mining is that we have something on the order of 20,000 kilometers of streams that are impacted by acid mine drainage from mine sites. Now, if we go to the Morenci copper mine in Arizona, the USA, this is the Metcalf pit. The Morenci mine is producing 1 million pounds of copper per day with bioleaching. So these are the same bacteria that are causing acid mine drainage from the mine tailing. And while the leachate from the mine tailing is uncontrolled and they're viewed as bad bacteria, at the Morenci mine, the bioleaching of mine tailings is attributed to good bacterial processes. The chemistry, though, is identical. It just comes down to whether we're controlling the leachates from our mines or leaving it to impact our environments. So if we zoom into this little circle here in the middle, we find a fracture where you've got abundant iron oxides. We've got chalcanthite, a copper sulfate. If you go into the lab and open a copper sulfate bottle, it will look blue, just like we're seeing in this natural environment. And what's remarkable here is that you can't make chalcanthite unless you have 60 grams per liter of copper. So it's a high copper mineral. And basically, anytime it rains here, it will just dissolve and then be recovered by the mine. So it's not being allowed to leach. And when we screen for iron oxidizing bacteria, we can find our iron oxidizers. When we screen for sulfur oxidizing bacteria, we can find our sulfur oxidizing bacteria. And both organisms are producing acid as a byproduct of metabolism. And so if we can exploit this ability to generate acid and to leach metals, then that would be better. than allowing acid mine drainage to occur. Now, our interest is increasingly going to critical metals. So cobalt and nickel, which will be important for our batteries, always the precious metals, the platinum group elements, and then the rare earth elements down the bottom that are critical for magnets and our decarbonization efforts. We'll take a look at a cobalt example here. This is the Princess Creek tailings. You can see the rustiness is starting to build on it. It's generating acid mine drainage. And for the last decade or so, the Princess Creek tailing has been viewed as an acid mine drainage site and being caused by bad bacteria. And the reason is because it has 7% pyrite, and pyrite or fool's gold is an active substrate in generating acid and causing AMD. And even though it has some iron, it isn't an iron source. It's not economic, and so we've had leaching going on, causing a problem. The other thing about the Princess Creek tailings that was never targeted from a mining perspective was that it also contains cobalt within the pyrite. And so if we can use acid generating bacteria to break down pyrite and to extract cobalt, then we would have changed these organisms from being bad organisms to good organisms. And when we look at the surface of the mine tailing here, we've got our iron oxides, so the evidence of iron oxidation going on. The unoxidized material are gray and are reduced. And so we're looking at the bacteria now within these environments. Can we use them to extract cobalt? We've been going to the Australian synchrotron to look at copper and zinc within the system. Cobalt is proving a bit of a challenge because it overlaps with the iron signal. So we got a good iron signal here. We need to go back and do speciation mapping to get at the cobalt signature that's in the environment. One of the things this image also highlights is that there's a lot of copper within these environments, and the chalcopyrite in mines all over the world are still a challenge in bioleaching. And so we're looking at unwrapping that ability, because we do get copper mobilized within this environment, and we have to improve our understanding. So if we go from this oxide cap here, a little deeper in the mine tailing, we can find copper and zinc and a little bit of iron that's becoming enriched. And so this ability to precipitate or potentially recover copper, zinc, and ultimately cobalt is the challenge in mine tailing. So can we take this waste and turn it into a resource? The exploration geologists, right? They're always critical of the process people that might have 90 or 95% recovery. In some cases, it can be as low as 60% recovery from some mine sites. And so the explorationists comment that it's really hard to find these deposits. And once we do, you should make a better job of exploiting them. And so that's what we're trying to do in this within this system. So let's go to Brazil. So this is the rainforest near Carajás and Pera in Brazil. And if you fly up onto the plateaus within this environment, we can go from rainforest to scrubby, dry plateau. So you're still in the middle of the rainforest. The inset image shows this feature. It's on kilometer scale. And these are the iron deposits in Brazil. And if you dig into this material here, you'll see something like this. We've got basically a 5-meter layer of rust that is protecting the ore below from washing away. And as you go deeper, this original banded iron formation has had most or all of the silica washed away, leaving pure iron. The latest mine that they built in Brazil does not involve trucks or blasting. They're just digging up this soft ore and putting it on rail cars and taking it to market. So this rust is responsible for the ore deposits forming, and we're interested in forming this rust because this is the natural habitat that started before mining. And if we want to rehabilitate the site, then we have to be able to reproduce this kanga or this iron oxide from the top. If we zoom into it, I typically describe it as an organic looking rock, and that would likely make Evelyn cringe because we don't have organic rocks. But it looks akin to the stromatolites. We've got this layered structure that grows within this environment. If we take that and polish it, we can see our layered structures. And if we zoom into this layered material, we find that it's absolutely full of fossils. There was a geologist, Dorr, in 1964, who was studying Kanga, and he said it was so weird. There had to be biology involved in some capacity. And what we've shown is that there are bacteria throughout the Kanga. So if you fossilize the bacteria with iron, you can make, in this case, a 5-meter layer of cement that protects the ore below. We are also able to find, hiking around the edges of the Kanga, areas where we had groundwater interacting with the rock and seeing it actively forming today. So as the iron is dissolved by the addition of organics and bacteria and flows out, as it encounters oxygen, we form the iron oxides that produce these layered structures. And if we just we took a test. So on the top left, you've got an IBC there. So you're looking at basically a meter squared face of a Kanga ecosystem. Once we'd added our bacteria and let things grow, the entire cubic meter became cemented with itself. And if we zoom in, we can see layers of iron oxides, some of them very brittle and gentle, but others much more robust, acting as a cement or glue. And so we dug throughout the whole pile, trying to find areas where things had aggregated as a demonstration that we can reproduce Kanga. And so this is our hope, is that this would be a rehabilitation strategy. Once mining is complete, we can grow Kanga and protect the material from washing away. From a mine tailings perspective, it would be much more challenging. But if we can cement mine tailings, then hopefully we would reduce the risk associated with mine tailings disasters associated with iron mining in this case. So here's an experiment from the lab. We've got our big class of kind of crushed rock that was left over. And what you can see is that there's kind of a darker gray connection between all of these materials. And if we zoom into that, here's one material that's it's almost acting like a suspension bridge, linking these two materials together. And to zoom in and take a closer look, it's like a bacterial sponge. So it's full of fossils. The student was concerned about the sample breaking and being damaged and not being able to analyze it. In actual fact, by recycling as little as 2% of the iron, the student needed a chisel and hammer to break it apart in order to process these fossils. So these biological iron cements are incredibly strong and should hopefully be able to help remediate iron mine sites. About 15 years ago, a colleague from UBC, Greg Dipple, invited me into the field with him, and we went to Atlan, BC, that had this hydromagnesite deposit. So a natural environment where we were sequestering CO2. We got down to the surface of the hydromagnesite, and as Greg started into his talk on the deposit and how it might have formed, the microbiologists in the group became distracted. And if you look off to the left here, we've got a pond. And the reason we became distracted is that the pond had microbial mats. And microbial mats are active in mineral carbonation. And while he was keen to look at the hydromagnesite, we really never went back there. And we started looking at these films. If we zoom in on the mat, we get a nice close-up. They're almost centimeters in scale. They're fixing CO2 into biomass. But cyanobacteria will also take in bicarbonate, fix the CO2, and pump out hydroxyls. And the hydroxyls there are critical for mineral carbonation, and so represent a natural attenuation process to our carbon emissions. If you put it in an SEM to get down to the bacterial scale, the oil barrel-like material there, that's our cyanobacteria. It's covered in heterotrophs and exopolymer. And when you add the magnesium and the leachates from Monarch Mountain in this environment, it's actually old ocean floor that's weathering, and so it has calcium. It's rich in magnesium, and when we add that to our reaction system, you can see the mineral plates that are forming in the carbon polymer around these bacteria. This is our hydromagnesite, so we got a magnesium carbonate that's forming. One thing that was remarkable is that biofilms also produce a lot of exopolymer. And so if we look at the exopolymer within these microbial mats and biofilms, we actually saw that minerals were growing in this spiderweb-like texture of EPS. And EPS is about 5% solid and 95% water. And so you can imagine before it became dried for the SEM that these minerals would have formed within a gel. And so it's this gel that has a net negative charge that brings in cations like calcium and magnesium, and then the photosynthetic activity, which shifts the chemistry towards minerals that are forming. Probably one of the most interesting images that Janine McCutcheon produced is shown here, where we've got a massive amount of exopolymer that's nucleating the growth of the minerals. And so I would argue here that what we're looking at is a biogenic mineral. It's the EPS by the bacteria that are nucleating the growth of these materials. Now, being at the University of Queensland, we have half of an island, Heron Island, in the Great Barrier Reef. So the front edge is a resort, and if you're interested in diving, the reef here is still in excellent quality. The other side of the island is run by UQ as a field station. And islands in the Great Barrier Reef have beach rock. And beach rock is attributed to protecting these reef islands from erosion and storms and loss of material. And we were interested in the beach rock. The geologists had claimed it as theirs. So if we go down to the beach rock at low tide, it's completely covered in materials. If we look at the same kind of section of beach rock at high tide, you can see the beach rock that's in there. You can see areas where beach rock is broken or collapsed, and you've got layered structures off to the left that can show the kind of the growth rings on this island. One of the fascinating things about beach rock is that these cements are self-healing. That you can see a couple of arrows there looking at, kind of younger material that's cementing the older clasts where there's been some damage and collapse from storms. And so we were interested in this self-healing. There's a lot of talk in bioconcretes that can be self-healing and self-repairing. And so we've got a natural example of this in this environment. If you look at the surface of the beach rock, we've got our classic biofilm, just like we were seeing in Atlin, British Columbia. And if you zoom in, you can see the close-up of the biofilm. We've got photosynthetic organisms. We've got oxygen bubbles that are being produced that are showing us that where photosynthetic activity is really active. And if we start digging into this material, we find that it's full of our bacteria. So we've got our 3D view of our cyanobacteria that generate the alkalinity. We've got the EPS in this environment, and we've got a wide range of bacteria growing on the surface of beach rock. Now, if you treat it as a geologist would, you'd take the rock, you'd cut it, and you'd polish it, and you'd take a look. And so we can see that we've got a calcium carbonate sand from various marine organisms, from mollusks to coccoliths within this environment. And we got kind of draping textures of biofilm, but the geologists don't really think the biology is too important here. If you zoom in, we find that the cements are growing by aragonite needles. And so the geologists would argue that seawater is saturated with aragonite, that splashing and wave action degasses, and that this was evidence for an abiotic formation of beach rock. But if you process this material as a biologist would, we find that it's actually covered in exopolymer. So we've got a soft, zoomy coating on the aragonite here, and zooming into those aragonite needles, we find that it's covered in exopolymer. So in nature, it would be a gel. It would be covered in EPS. And so if the hydromagnesite is a biotic mineral, then the beach rock here forming an EPS also has to be considered to be a biological feature.
Gordon, just quickly, for those of us who are not microbiologists, what is an exopolymer?
Sorry,
just define what an exopolymer is for those of us who are not microbiologists.
Yeah, so exopolymer can be something as simple as capsule. So it's a carbohydrate. So it's a high atomic mass polymer. Have you ever been hiking and seen like there's like little seeps or water that, and if you stand on it, you slip?
Yep.
You're slipping on exopolymer. So it's that highly hydrated gel that bacteria grow in. It can, if bacteria are growing in an environment with toxic metals, the negative charge on EPS can help protect against those metals by binding them. In terms of pathogens, they will tend to grow as biofilms, and so it would be EPS. So in pneumonias and things like that, the EPS and damage that's caused to cells help protect organisms from their environment. It provides a different chemistry and different structure for these things to grow. And when you look at this here, like you can see, we're down to micron scale to see these bacteria. that if you get to millimeter scale, right, and you just personify it for a sec, that if we were the size of bacteria, then a millimeter scale biofilm to us would be a kilometer in thickness. And so you think of the changes in chemistry and things that can occur over a kilometer scale. Down the Gold Coast here, it would mean the difference between being in my office and being in the ocean, right? So you get different chemistries and different environments that are exacerbated by exopolymer, allowing gradients.
Thanks, Gordon. That's helpful.
So here's where we met, Evelyn, was you setting up a workshop in South Africa to launch this project. And I like this one because this is the De Beers media release associated with can we produce a carbon neutral mine? And the press release that we saw from it was something more like this, right? De Beers to make carbon neutral mining a reality across its operations in 5 years.
Yeah, and I talked to the media team about this release. I protested it and I said we shouldn't put any time frame on it. We should just say it's possible, because at the time they released it, it was really sort of research. So
yeah. Well, and also it took us a year to get on site.
It did. Well, we got affected by COVID. Anyway, I'm sure you'll talk about it. But I think sometimes it's challenging. You obviously want to get corporate support for these things, but you also need to make sure that you've got sufficient time to do rigorous scientific research, and those two things can sometimes play against each other.
Well, the exciting thing to me was the scale that we got to operate on. So we're looking at mine tailings. So we're getting onto mine sites, and that we also got, we're similar to the Brazil projects. We're getting into the cubic meter scale, which from a bacterial perspective is on the order of 9 orders of magnitude, right, from the cells themselves. And so cubic meters on up is a challenging space to look in. So I was inspired, associated with this project with yellow ground from the Carivale mine. So this is weathered kimberlite and has a massive calcium signature associated with mineral carbonation.
And maybe just to take a step back for those of us who are not geologists. So kimberlite is one of the rocks, the most common rock that diamonds are found in. And if you look back historically at the mining of diamonds, the early days, so when they started to really have a lot of production from Southern Africa, particularly South Africa, all of the early diamond mining was in this rock that they didn't even know it was kimberlite initially, but it was just called yellow ground because it looks like this. It's really weathered. And this rock was quite soft. It could break it apart in your hands. It was really easy to crush and produce diamonds. And eventually, after those early days, when they got through the yellow ground, they realized that the yellow ground didn't go down forever. And then they encountered something called blue ground, which was the same rock we found out years later with scientific research, but just much less weathered. And actually, a lot of the early diamond mines, they actually stopped for a long time period because they were not able to economically at that time process the blue ground that was underneath. So if you're trying to figure out how you can get your blue ground, which is what we typically mine today from diamond mines, to weather and to take up carbon when it does that with what we're interested in, it's really interesting to look at this yellow ground and learn lessons from it. And Gordon was interested in knowing the microbiology of what's creating and living in that yellow ground.
One of the interesting things at that time was that a lot of the diamond fields in South Africa were literally fields that they took blue ground and spread it out, right, to promote natural weathering. And in as little as a few years, it would have weathered enough to get the diamonds. So this was, kind of the target for us, right? Could we produce something along these lines? So here's the Venetia diamond mines. We've got biofilm growing within the mine pit. They're full of photosynthetic organisms, and so we were targeting these organisms within this environment. So if we zoom in here, we've got 1,000-liter culture vessels. We've got our microbial mats. We had a real challenge though, is how do we inoculate the kimberlite with this biomass that we grew from the mine site. And the solution proved remarkably easy, that bacteria tend to stick to minerals and minerals will stick to bacteria. And so we just shoveled finely crushed kimberlite into these bioreactors, and everything just kind of produced a cement and stuck together. And you can see the IBCs in the background there, where we shoveled the material to sort of start running our experiments. So here's our coarse reject. So it's kind of 0.8 millimeters to 8 millimeters in size. We've got class. This is our coarse residue from diamond mining. This is the crushed kimberlite, and this is the control. And for the entire experiment, up to 4 years, it just looked like this. What was remarkable was that in the biologically oxidized one, our CRD Bio, that the kimberlite had changed in such a way that it started functioning as a soil. And you don't make a soil from primary material unless you weather it. And so we had weathering reactions here. Now, we didn't plant anything. These are plants that literally had seeds that just blew into the mine environment that we were working in and colonized kimberlite. And so we were excited because we had weathered kimberlite and would it be like the original materials. So we needed trowels. In some cases, we need a hammer to break into the surface where we had the plants, that all of the material had started to become weathered, similar to yellow ground, and was cementing itself together. If we zoom in there and start getting down to millimeter scale, we found our calcium signal. So we're producing almost tiny stromatolites that are full of bacteria that are sequestering carbon. In terms of the amount of carbon that was sequestered in these bioreactors, it was on the order of about 20% of mine emissions associated with that cubic meter of material. And so this aspirational goal of producing a carbon neutral mine is not subject to geologic time, where you can have thousands or hundreds of thousands to millions and billions of years, that in actual fact, it's on the human life scale and on a mining life scale that we can sequester significant amounts of carbon. The latest project that we have is last year I was part of the International Ocean Discovery Program to drill the Atlantis Massif. So the Massif is a 4,000 meter tall mountain at a tectonic spreading center in the middle of the Atlantic Ocean. And in this environment, we were able to drill down through 1.3 kilometers of peridotite. And peridotite, like kimberlite, is an ultramafic rock that readily weathers and so should be a target for carbon sequestration. And so here's a glimpse at one of the samples we have. We've got carbonate veining. A student has now extracted DNA from this material, and we're excited to go look at it with high resolution microscopes and the synchrotron later this year to get a better understanding of how carbon is sequestered within these ultramafic materials. Last topic of the day is gold. That everyone should know how to grow a gold nugget at the end of this section. So this is a gold grain. It's from West Coast Creek. It's placer gold, so it's the stuff that you can find by panning near Kilkeven, Queensland. And so you have a little Eppendorf tube there on the left with I don't know, probably 50 or 60 gold grains that we recovered from this environment. We're not quite to the scale of bacteria yet, but we're getting there. Now, if you look at this with this image is with secondary imaging, which gives us a 3D view of this material. If we go to backscatter, then we get contrast based on atomic mass, and that high atomic mass things will be bright, and low atomic mass things will be dark. And so if we shift the detector here, we've got a couple of circles in the top left. We've got dark material, that's soil or sediment from the creek where it was isolated. And we've got tiny little particles of gold. And we'll come back to that in a little bit. With the other circled more towards the middle of the grain, we've got bits of gold, and then we've got kind of a patchy texture of larger particles in this environment. If we zoom into that environment, we can see that we've got gold that was described as bacterial morphic. And just highlighting the fact that it kind of looked like bacteria. We've gone in and looked at these materials, and if you cut them open and look inside, they're not actually bacteria. It's just a dissolution or precipitation feature associated with gold weathering. And this idea of gold weathering is remarkable, right? You can whether you've got jewelry or rings or something that's made of gold, we tend to treat gold as something that's inert, right? That it doesn't weather, it doesn't break down. And what we're seeing when we look at these natural materials is that there's lots of evidence for a gold cycle. We've seen evidence of an iron cycle and sulfur cycle and copper and nickel and zinc and associated with mine environments, but gold is an unusual target. And so this is unusual that there's evidence for so much gold dissolving and reprecipitating, that there's an active gold cycle out there. If we zoom into the soil in this environment, we find that it's loaded with crystalline gold. And the circle here highlights a grain of octahedral gold. What you're seeing in plan view is the 6 sides of a hexagon plus the top and bottom of that flat crystal gives us our octahedral shape. And so within the soil, we've got colloids, we've got tiny particles, and we've got crystalline gold. And the crystalline gold in these natural materials was really exciting to us because we can make crystalline gold in the laboratory. So here's a cyanobacterium, Plectinium boreanum. When it's exposed to gold chloride, it precipitates the gold, it forms colloids. You can see little bits of exopolymer that's in there. And over time, we produce crystals that are microns to even tens of microns in size. In between the patches of gold, we find that there's bacteria throughout the soil. And so we've got bacteria here that are growing. You can see the contact between the bacterium and the gold in the top right is very sharp. And so we've got this dissolution process that's going on. And geologists have known this for a long time, right? That when you have a rock that contains gold, you often find dispersion halos. The idea that gold is being weathered, and in some cases, the gold signal will disappear and reappear downstream. And so we're interested in that because what we think is happening is the bacteria are weathering the gold, they're dissolving it, and then they're growing gold crystals. And so this idea, not only the crystals, but we're growing gold grains as we go downstream. So here's a kind of a composite image of a series of experiments that we've done with gold. Now, when we do experiments with iron or copper or zinc, when we're done with the experiment, we dispose of the materials, right? It's not viewed as having value, and we clean up and we continue on with our experiments. Funnily enough, students working with gold, when they would finish their experiments, they wouldn't throw them away. I said, "There's gold in here. You can't." In good conscience, they couldn't chuck them or get rid of them. And so what we had was a closet in the back corner of the lab that over a decade was basically becoming packed with gold experiments. And so we have in the top left evidence of bacteria precipitating gold along the shape. We've got a biofilm of gold. We've got our octahedra that cyanobacteria produced. And these biofilms of gold represent that intermediate between weathered rock and growing a gold grain. Now, we tried to think of ourselves as geologists, and this was the best we could do. We got a tumbler. This was Jared's tumbler from Christmas in grade 3, and he brought it to the lab, and we took our gold experiments and tossed it in the tumbler and plugged it in. We let it run for a month. And one of the things we retroactively thought about was how long or how far had these gold grains traveled within the tumbler. And it turned out to be 80 kilometers. And surprisingly enough, in nature, that if you look at hypogene material or weathered gold grains, they can actually over tens of kilometers begin to grow. And when we opened this bioreactor at the end of the experiment, this is what we found, that we had in fact produced gold grains. If we zoomed in, we had our octahedral gold crystals. And so it is possible to grow gold grains. And we think that it's intimately associated with bacteria. And with that, I'm close to my 45 minutes, so I'll stop there.
Thanks, Gordon. I didn't know you were going to end with alchemy and I guess your new funding mechanism for research potentially, although it's 10 years of experiments. That's pretty cool. So we'll have some time for questions in a minute. I realized that I think I missed recording the first couple of minutes of the talk, Gordon. So if you don't mind, after the questions are finished, if you could just stay on for a minute, we'll just go back to the title slide and we'll record that just so that the video at least has the beginning bit. And then the alum office at MIT, I'm sure will work some magic and do some editing and put it together in a reasonable fashion. But apologies for that. So that was, for those of you who missed it and joined a bit late, I did have to send the link out to a couple of people who didn't get the email correctly from MIT. Gordon has many, many years of experience working in geomicrobiology, looking at microbiology and interaction of microbes with minerals. And what he did today was give us a tour through not all of his research, but I think just a selection of some of his recent research. So that was really good. My head is spinning a bit, but I would love to open it up now to the group if anyone has any questions for Gordon about the talk or about anything in the geomicrobiology field.
I have a quick question for Gordon. I was curious about, you had mentioned when you were discussing the De Beers mine and the yellow ground, when you were trying to extract the mineral bacteria adherents you talked about. I was curious about that. What can you elaborate a little bit on the chemistry behind that?
Yeah, so fundamentally, if you look at all of these projects that I kind of highlighted today, I'm interested in bacteria mineral interactions. And so those interactions can be, one where we're dissolving minerals. And in the case of acid mine drainage and bioleaching, so that's an example of degrading minerals. And equally important, bacteria can be involved in precipitating metals. So, and forming new secondary minerals within those environments. In the case of, in the case of the mineral carbonation experiments that we were doing with Evelyn, we've got an environment there where the photosynthetic bacteria and there's other heterotrophs that are acting deeper. Basically, we're looking for the ability of biology to enhance weathering. And so bacteria will produce a lot of organic acids and inorganic acids that can degrade materials. They do this when they're looking for phosphorus. That phosphorus within fertilizers has been treated with acids, but in nature it tends to precipitate as a calcium phosphate. And so plants and bacteria and fungi will produce acids that help break down apatite and release the phosphorus that can be used to build cells. This weathering process in kimberlite will release calcium and magnesium. And so we found the most activity to be near surface. And that makes sense because cyanobacteria in these environments, that in an alkaline environment, it won't tend to have CO2. And the CO2 dissolves from the atmosphere. It's as a bicarbonate phase. So you've got an HCO3 minus. And what the bacteria do is they take the CO2 from the HCO3 minus that leaves you with the hydroxyl.
Okay.
The hydroxyl drives up the pH. The pH makes carbonate minerals, just like you see in the Great Barrier Reef.
Well, I was more around the mixing. Like, how does the bacteria get with the mineral when you put them in that solution? Is that what you were after?
Well, so I guess, the bacteria will become entombed. Right, so it's often described as permineralization, where the minerals are precipitating so quickly that they're trapped inside. And when they're trapped inside, they'll have lipid, they'll have protein, they'll have DNA. All of the biochemical guts of the organisms will be preserved within that rock. And when we come along, we will crush the rock and extract DNA, whether it's the kimberlite from South Africa or the weathered ultramafic material from the ocean drilling program or bioleaching systems that there's remnants of bacteria, but they're biomarkers, right? So we don't just need cells that if we can find, kind of DNA sequences that tell us about what's living there, then that's a bonus.
Sorry, I interrupted you. Go ahead.
That's all right. No, I was just fascinated because I wish we would do similar type analysis within us with our microbiome, because I suspect, when we put different people on different mineral supplements trying to replace deficiencies, I find there's such variation in how much people will absorb. And I'm suspecting it has to do with their microbiome components and distributions. And it'd be great someday to be able to take a biopsy of the esophagus or the small intestine and do this similar type analysis to figure out.
So one of the coolest ones we have at the Australian synchrotron was they had a sample of Farlap's hair.
Oh, wow.
What they showed was that over the last, kind of weeks to months, that the amount of arsenic in the horse was increasing and was being, and so they found it and could measure that arsenic exposure.
That is fascinating. Yeah, really interesting.
Great. Do we have anyone else with a question for Gordon?
This is not so much a question, but as you commented, Evelyn, my head is also spinning, and I'm doubtless coming from a lot lower knowledge base in this area. So I was wondering if Gordon could just suggest a reference or two that might just help bridge to the content of the lecture, because I'm certainly keen to rewatch the recording as well.
So there's a series of elements articles that are out there. I could send to Evelyn. Is that,
if you send those to me, I can email those to you.
Yeah, that'd be great.
So
Yeah.
I also recommend that. So Elements Magazine, you need a subscription, but if Gordon's the author, he can send it to you.
Yeah.
And that's a really nice magazine because it's geared toward non-specialists. Because even for me, I'm a geologist, I'm a geochemist, but I think working with Gordon has really opened my eyes that a lot of the reactions that I learned about at MIT as being inorganic reactions. We're realizing more and more that they're not inorganic at all, they're organic. And we don't like to think of our rocks as being organic, but many of them probably really are. Yeah. So it's something that is interesting because I think it challenges a lot of what we know. And I think where it often plays a role, or where increasingly scientists are trying to apply it, is with kinetics. So can we leverage these microorganisms to help us take something that, if we don't leverage them in the right way, is going to take a really long time, and it's something we want to speed up? I've mostly worked in CO2 drawdown and carbon storage, but there's loads of applications like dealing with that acid mine drainage and solidifying things, all sorts of things where you could try and design a quite expensive and often a process that has a big environmental footprint that uses traditional chemicals. But if you could use microbes, you might be able to do it more cheaply and faster and without a lot of the environmental kind of impacts that you might have by trying to do traditional chemistry. But yeah, there isn't necessarily, I'd say up till today, even a good teaching of this in kind of your basic geochem. And I think that's maybe starting to change, Gordon. But those Elements articles are a great place to start.
I have a question.
Go ahead.
Can some of this biology help us with surveys for discovery?
So I have a chapter that I wrote for a book called Heavy Metals, and in that there's increase. I've got molecular biology colleagues. UQ has a center for molecular biology that are or the Australian Centre for Ecogenomics. And so metals, Simon Silver years ago, a microbiologist interested in metals, basically, said that bacteria could transform, every element on the periodic table. And he didn't have proof for it yet, but as we gradually work through, we see increasing evidence for bacteria using anything. Even kind of rare earths, lanthanum is used in a methanol monooxygenase. So this idea of metals as cofactors in bacteria are critical. If you look at the number of genes associated with bacteria, and we've started looking at mining environments, at the regolith above a mine, can we find genes that target specific metals? And there are hundreds of genes for copper or nickel or zinc. If you look at the metagenome, there's actually thousands of different genes that might suggest that there's ore below here. So can we work with conventional geochemistry to add another tool to the kit to say that there's genetic material down there? And the advantage of the genetic material is that bacteria are very good at holding on and storing a lot of genetic material just in case they might need it one day. And so where geochemistry will capture, kind of the chemistry of the day, the molecular mechanisms or the molecular biomarkers that are in there are going to capture the kinds of chemistries that these organisms have seen, basically for geologic time.
That's fascinating. Thank you.
Great.
I can share that one too, Evelyn. Yeah.
Sure. Yeah, send that to me and I'll make sure that gets sent on to you, Ted. Are there any other questions about the content of the talk or just in general? got plenty of time. Well, if you think of something, drop me an email and I'll put you in touch with Gordon. Thank you so much for coming to the talk today. Gordon, if you'll hang on for just 1 minute, we'll just re-record that intro. And I will see you all at the next talk. One of the next ones that we have planned, I should get it advertised next week, is I have a colleague, she studied at MIT, and she's had a career in medical engineering and has worked for a number of different companies. So that should be a really interesting one. And then coming up in a couple months, we'll also have a talk or two on AI, which I know is at the front of a lot of people's minds at the moment. So there'll be some interesting talks coming up. And if anyone here would like to give a talk or can suggest a good speaker, please drop me an email. We'd love to get a few more people on the calendar for later in the year and into next year. Thanks, everyone.
Thank you for your organisational efforts, Evelyn. So much appreciated.